All right, so I guess it's time now to start. So once again, thank you for joining us today. We are talking today about how high content imaging is driving the early drug discovery pipeline. And basically, your host of this talk and the presentation will be done by myself and by Dr. Stefan Prechtl. And before we start, I would just like to give you some 2 to 3 sentences about ourselves. So myself, I'm a trained chemist, but I love to basically work on image-based assays since already my PhD, since I was able to tag, for example, proteins or organelles using fluorescent probes. And this did not really went away during my pharmaceutical development. I was having research positions at the German Cancer Research Center. Then I matured more in cell-based screening strategies, but also in high content imaging and complex cellular models during my activities at Novartis in Basel. And since beginning of the year, I'm now complementary adding with my expertise to the Nuvisan Innovation Campus, Berlin. And now I would like to ask Stefan to introduce himself.
Yeah, good morning, everybody. My name is Stefan Prechtl. I'm a biologist and I did my PhD in immunology a couple of years ago. I've been working in industrial pharmaceutical research now for a little over 20 years. During that time, I was working for Schering, Bayer, Nuvisan ICB also, but in January this year, I started working for my own company, which is called the CELLIMA Scientific Services, GmbH. And with the CELLIMA, I am offering consulting services, training in high content analysis, high throughput screening, cell biology, and microscopy. And it is a great pleasure for me today to be part of this webinar together with Florian and to support Nuvisan, my role as the external consultant, to introduce to you this fascinating technology of high content analysis. Together, we both will offer you a deeper insight into this fascinating technology and show you how it can be fully utilized at the Innovation Campus here in Berlin.
Thank you, Stefan. So what is the agenda today? So we would like to talk about high content imaging drives the engine of early drug discovery, as I've mentioned. And during today's talk, we would like to provide you a small insight into the services that we are offering, and then come to the main topic of this presentation, how high content imaging has evolved over time, what requirements are actually needed to do this successfully for early drug discovery research, how we are using it in Berlin to drive our preclinical activities, and which future challenges we would like to tackle on. And of course, we will be ready to take your Q&A session basically afterwards. So let me first introduce you into certain house rules during this talk. So please be aware that your microphone is muted by default. And of course, you can use the Q&A chat box to ask your questions that come along this presentation. And we will be very happy to try to answer them in time or afterwards, after our presentation, of course. You will get the recording and the slides after the presentation as well upon request. Now let us start with the first poll. The question is, have you worked with Nuvisan before already? It'll be quite interesting to see the answers because we have received registrations from all over the globe. So it will be very fascinating to see how you are answering regarding this poll.
Did you get the whole globe, Florian, or is there a specific dedication to one of the, let's say, regions?
We actually got the whole globe, to be honest. Cool. Maybe New Zealand is missing yet, but maybe afterwards. Here's the answer. I'll shift it a little bit further. One moment. Oh, that's interesting. So
yeah.
a huge majority of you have not worked yet with Nuvisan before. So I hope that we can change this after this presentation, obviously, and that's what we are trying to do now. So sorry for this, I just need to. So Nuvisan, if you haven't heard about it yet, is a fully integrated conflict research organization. And we basically can offer everything from your crude idea, from having an understanding of a target that you would like to work with regards to drug discovery purpose, to a proof of concept in patients, meaning you get, if you want, a package delivered from us, from your target to the patient. And as the Innovation Campus Berlin, we are actually, myself, residing, for example, within the lead discovery unit. So this means we are taking care, together with our other functions here in Berlin, from translational research, for example, not only to do the target identification, but also the validation. We screen using various methods, and high content imaging, as you will see, is one of the biggest assets we have here in Berlin to do so. And together with our chemist, we are able to also optimize the lead candidates that we have identified and aligned on, and also characterize them to the very extent that is needed to drive it further into the preclinical trial, which will be executed upon need together with other Nuvisan parties located elsewhere, but not in Berlin. Now, high content imaging is, as I mentioned already, one of the most versatile readout technologies that we have available during screening. And we use it not only for screening of large small molecule libraries, but also of subsets thereof, depending on your particular interest you are looking into. But besides screening of these large small molecule libraries, we can also offer optimization studies, functional activities to identify a mechanism of action or something like this. So really, we are using it all over the whole preclinical stages that we are able to execute on. And the main benefit of running high content imaging assays is that it is not only a complementary add-on to, for example, biochemical or biophysical studies, but it is due its richness in information, absolutely a strategic fit into your screening proposal. Now I would like to ask Stefan to explain a little bit how high content imaging started with regards to readers that were used and how this was explicitly set up at Berlin.
Yeah, thanks, Florian. Let's dive right into the topic of high content imaging and have a look at the different applications. And this is a good point to start what we call with the evolution of high content analysis. This largely depends on the evolution of the technologies that are involved and are necessary for successful implementation of scientific ideas with regard to microscopy, for instance. Particularly important place here is occupied by the measuring instruments, and without them, an experiment would not be feasible to perform. Automation is key for industrial research, and this applies equally to the development of high content imaging technology. And microscopic work was possible to automatize for the first time when the motorized microscopes came into place. But these devices were optimized for human access and still required a great deal of manual intervention. The next step in the evolution was to develop a fully automatized microscope. But these were still just converted motorized microscopes that were more or less hidden within a plastic box. And since these devices still were originally designed for manual operation, they required a cumbersome modification on the hardware side as well as on the software side in order to be able to perform at least medium throughput capabilities. And a relatively high level of monitoring was also required to ensure that these devices could reliably perform small tests. With the next generation of high content readers, the developers have then applied true functional design. And for the first time, process optimized devices were produced and available, and these devices were able to work 24 hours a day, 7 days a week. And thus, for the first time, large primary high content analysis screening was feasible, even though to that time the entire screening process per se took significantly longer when compared to standard biochemical screening or reporter gene cell-based screening applications. With the second generation of these devices, which have been around now for a few years, there has been again a significant improvement in these devices with regard to resolution, throughput, options of individualizations and applications that can be applied to the systems. And these devices have also a smaller footprint now, and they can be easily integrated into complex robotic lines because they have optimized interfaces in place. They are efficient and reliable, and they now offer primary screening, which is has an identical throughput than compared to biochemical screening approaches. But what about the assay, Florian? How do they evolve over the years?
Well, the assays, okay. So, I mean, the very earliest assays that have been performed using high content imaging, they were also relying on, let's say, similar to reporter gene-based assays, on rather simple labeling procedures. So they were focusing on maybe one aspect that you would like to take as a readout parameter, for example. And counting was pretty easy and very efficient on these kind of protocols. Nevertheless, these protocols are still sufficient to answer certain of your biological questions you have in mind. And it's very well, it's good practice to use them still in these days if they would provide you with a physiologically relevant answer. Now, multiplexing of fluorescent dyes or probes was becoming quite after very soon available. And this was basically also used and can be of good value, very good value for you actually, if you're using those multiplexing in a screening setup and use these gain of knowledge to have a higher confidence in selection of your candidate hits. Most of these standard 2D cell culture models in these days are actually being performed in a multiplex readout, for example, as endpoint assays. If you combine the multiplexing of your markers with the biological information and use particular biomarkers to address these functions to elucidate signaling pathways, for example, you can even get more information out of it. I personally got always excited when I do not only see an image, which is already saying 1,000 words, as we know from many examples, but when I see sequences of images, when I have the ability to understand how something has evolved over time. And this was becoming a real game changer for me, where I got introduced to in Basel, and where I'm really happy to see that this is happening in Berlin now still as well. So we can have live cell approaches to understand how a cancer cell, for example, is dying because it is not able to form any longer a bipolar spindle, how certain cell types are getting phagocytized and in what dependencies. This is quite amazing still in these days. Last but not least, we do have also even more advanced cell models, and these advanced cell models, they are trying to take into account that cells are interacting with each other, that they are exchanging messengers, that they try to have some other ways of communication, that they are influencing each other by stretching and so on. And that they also have to rely on 3D modeling, for example. And if you're thinking of human iPSC-derived cell models, for example, where defined differentiation protocols are needed to establish proper tissue connections, then this is quite exciting to be able to do this not only on a small scale experiment, but also to do it now in screening. But to do these kind of analysis efficiently, you really need to have advanced image analysis. And how did the image analysis evolve?
Yes, yeah, this is very important. After talking about developing assays and the evolution of readers who are capturing images, we have not yet taken care about the analysis. And if you want to have reliable image analysis in place, you need selected algorithms, image analysis routines, with you can quantify 100,000 or even 1 million images stably and